Hi, it's Dr. LeBlanc. This is our last video screen capture lecture in Unit 1, the molecules of heredity. In this unit, we're going to focus on the organization and packaging of DNA in eukaryotes. Our learning goals for this lecture are to understand the organization of functional sites on eukaryotic chromosomes, how eukaryotic chromatin is packaged, and how chromatin is organized in the nucleus. Eukaryotic genomes consist of a nuclear genome, which is packaged into chromatin and chromosomes. It also consists of mitochondrial and chloroplast genomes, which we're not going to discuss in this video lecture. A eukaryotic chromosome is one long strand of DNA from tip to tip. This is one long piece of DNA, unbroken strand of DNA from tip to tip of a eukaryotic chromosome. They can contain many origins of replication, approximately 100,000 base pairs apart. They have at the middle of the chromosome a centromere to which is attached a kinetochore, which is a complex of proteins that aid in making sure the two sister chromatids go to opposite poles during mitosis. They have telomeric regions on the ends of the chromosomes, which prevent shortening during DNA replication. And all of this DNA is wrapped around structures called nucleosomes. Nucleosomes aid in eukaryotic chromatin compaction. Nucleosomes are composed of an octamer, that means 8 histone proteins, and here is our basic structure. This is a nucleosome. These are the proteins, these orange globs. There are 2 each of H2A, H2B, H3, and H4. It's a dimer of 2 each of these proteins. The DNA gets wrapped around this ball of proteins, and then there's a linker region, and then another ball of proteins. Wrapping the DNA around nucleosomes creates a fiber that is 10 to 11 nanometers in diameter. I often refer to it as a 10 nanometer fiber rather than an 11 nanometer fiber. So a nucleosome consists of the 8 histone proteins and approximately 146 or 147 base pairs of DNA. The linker region in between the two nucleosomes can vary from 20 to 100 base pairs, depending upon the species and the cell type and the region of the chromosome that is being either transcribed or not transcribed. Here's a ribbon diagram of a nucleosome here. There are 2 molecules, each of H2A, H2B, and H3 and H4. And the DNA is wrapped around this. And you can see the relative structure or the relative size of the DNA versus the proteins. DNA is a very large molecule, and the proteins in the middle are a bit smaller. Also sticking out from the histone proteins, you'll notice these long tails. We'll get back to the function of these in future video lectures, but for now, know that these proteins' tails that stick out from the nucleosomes can be modified by acetylation, by methylation, by phosphorylation, and that those modifications will affect gene regulation. How is the structure of the nucleosome determined? Roger Kornberg, a very prominent DNA and chromatin biologist, proposed the model for nucleosome structure in 1974 based on his biochemical, X-ray diffraction studies, and electron microscopy images. Marcus Knowles' experiment, however, gave us a very visual and easily visually interpretable result to understand how DNA is wrapped around the nucleosomes. So his experiment began with nuclei, not extracted DNA, but actual nuclei. You can remove nuclei from cells and work with them individually in test tubes. So when you have a nucleus, you have DNA that is associated with proteins. These orange balls each represent a single nucleosome, and the blue line is DNA. He treated these nuclei with 3 different concentrations of DNAs. Now, his idea was that the nucleosomes are going to protect the DNA, but the regions in between the nucleosomes are going to be digested by DNAs. At a high concentration of DNAs, every single linker region of this chromatin would be cut by DNAs. But at low concentrations, very low concentrations of DNA, only some of these regions would be cut. Okay? That would leave different lengths of DNA at the end of the digestion. Okay, after he treated the nuclei with the 3 different concentrations of DNAs, he treated the nuclei with detergent and phenol to extract the DNA, and he ran the DNA out on an agarose gel. Agarose gels are a matrix. If you apply an electric field across the agarose gel, the DNA will migrate towards the positive pole because DNA is negatively charged. The smaller fragments will migrate quicker, and the larger fragments will migrate slower. So you can separate DNA based on its size by this technique. After the DNA has been run on the gel, you stain it with ethidiabromide, which intercalates into the double strands of DNA. It fluoresces under UV light, and you can take a picture. So this is how we interpret the data that Marcus Noll got at the end of his experiment. This is a length of chromatin. These are the nucleosomes, and these are the nucleosomal regions here, the linker regions. So when the DNA is cut by DNAs, you can end up with a length of DNA that is 1 nucleosome's worth of DNA. At high concentrations of DNAs, all the chromosomal DNA is digested into fragments that are 200 base pairs long. This is what the result he got. And this is how he interpreted the data, was that every single linker region was cut by the DNAs, and that the length of DNA that was wrapped around a single nucleosome was 200 base pairs. At low concentrations, however, not all the linker DNA was cut. You may have cut here, and then here, but nowhere else. So you ended up with fragments of DNA that were 1 nucleosome long, or 2 nucleosomes long, 400 base pairs, or 3 nucleosomes long, or 4, 5, 6, 7, 8, etc. So in this way, Marcus Noll was able to determine that the DNA in the nucleus wrapped in chromatin was approximately 200 base pairs long around each nucleosome. In addition to the core nucleosomes, nucleosomal proteins and an octamer, a histone octamer, there are additional proteins that bind to DNA in the nucleus. There is another histone called H1 that binds to the DNA just next to the chromatin, next to the nucleosome, excuse me. There are also other non-histone proteins known as, there are DNA binding proteins known as non-histone proteins that play a role in organization and compaction of the chromatin. And this is a very large group of heterogeneous proteins. Lots of different kinds of proteins are called non-histone proteins. Without H1, the chromatin is a 10 nanometer fiber. With the addition of histone H1, you create a 30 nanometer fiber. And the 30 nanometer fiber is a coil of the 10 nanometer fiber chromatin. So there are two different models, and they probably both exist in the cell. A solenoid model, which is very compact, where you have the nucleosomes wound tightly around each other, and a zigzag model, which is a bit looser form of chromatin, but they are both 30 nanometer fibers. Okay, so now we have our chromatin. We've gone from a DNA strand to a 10 nanometer fiber to a 30 nanometer fiber with the addition of H1. How do we get that 30 nanometer fiber to compact even further into chromosomes that exist in both the interphase nucleus and at the metaphase chromosomes that we see during mitosis? In a nucleus, there are many, many proteins. Nuclei are not an empty bag of fluid. They contain something called the nuclear matrix, which consists of lots of different types of proteins. Nuclear lamina, which is just under the inner membrane of the nucleus. There are scaffolding proteins, and there are matrix attachment proteins. This is an actual electron micrograph of a nucleus that has all its DNA removed. All the chromatin has been removed. And it's not empty. It has this matrix of proteins all around the inside. It looks a little bit like a sponge. This web of proteins still exists inside the nucleus, even if you take out all the DNA and the proteins that are bound to the DNA, like the nucleosomes. So just as prokaryotic DNA was organized into loops, eukaryotic DNA is also organized into loops. This green line represents a protein fiber of the matrix, of the scaffold of a chromosome. And the blue line, of course, is DNA. The loops can be quite variable in length, from 25 to 200 base pairs long. There are matrix attachment or scaffold attachment regions where the DNA is bound to the matrix or scaffold of the chromosome. And the Mars are anchored, the matrix attachment regions of the chromosome are attached to the nuclear matrix, creating these radial loops. So the DNA inside a nucleus of a eukaryote, although it may appear somewhat loosely organized, is actually quite organized. This is an electron micrograph showing a close-up of what the nuclear matrix looks like. Again, this has no DNA in it. This is just the nuclear matrix. Scaffolding of proteins that exists inside the nucleus. Chromosomes have different regions. They have heterochromatic regions and euchromatic regions. Heterochromatic regions are tightly compacted. They are at the telomeres, at the centromeres, and again at the telomeres. These regions of the chromosome are always heterochromatic. They are always tightly packaged, where the DNA is very tightly coiled around proteins. Euchromatin exists in the other sections of the arms of the chromosomes, and that's where we find the genes that are being expressed. The euchromatin 30 nanometer fibers are anchored in radial loops in these regions of the chromosome. The heterochromatin is constituted by greater compaction of the radial loops. This is an interphase nucleus, stained so that each chromosome is a different color. So here we have chromosome 1 here, chromosome 2 in green, chromosome 3 in blue. It's interesting to notice that even in eukaryotic chromosomes, the DNA, eukaryotic nuclei, that the DNA from individual chromosomes is not intertwined with other chromosomes, but they remain in specific regions of the nucleus. This is a little bit like the nucleoids we saw in prokaryotes. Okay, so this is a very important figure because it shows the hierarchy of organization of eukaryotic chromatin. First, we have the DNA double helix, which is approximately 2 nanometers in diameter. Adding the histone core, we get an 11 nanometer fiber. Adding histone H1, we get a 30 nanometer fiber. And furthermore, this 30 nanometer fiber is anchored by to form radial loops to the nuclear matrix. Here's a protein scaffold of a chromosome, and we attach these radial loops by the matrix attachment regions and form approximately 30 nanometer fiber here. You end up with further compaction of the radial loops when we end up with a metaphase chromosome right here. And the final diameter of a metaphase chromosome is about 1400 nanometers, excuse me. So euchromatin is loosely packaged, heterochromatin is more tightly packaged, and finally, the most tightly packaged you can be is a metaphase chromosome. If you take one of these metaphase chromosomes, however, and treat it with high salt to remove the proteins, you can see how the DNA is splayed out from the scaffold. And again, this looks very similar to the bacterial cell that we saw with its chromosome splayed out. That's a nice up-close picture of it. How do we get our chromatin fibers organized into metaphase chromosomes? There are two very important proteins, condensin, which plays a role in chromosome condensation, and cohesin, which keeps the two sister chromatids aligned. This is a model of condensin. Notice it's a very complex multi-subunit protein that compacts or organizes the radial loops. Cohesin is found in between the two sister chromatids. Initially, when the metaphase chromosome is not as compact, and even further along during metaphase, where the chromosomes are even more compact at the beginning of prophase. Finally, in the middle of prophase, the cohesin molecules are dissociated from the sister chromatids, except for right at the center here, at the centromere in the kinetochore, and that's the last of the cohesin to leave during anaphase, when the two sister chromatids split apart and go to opposite poles into their new daughter cells. So in summary, eukaryotic DNA is packaged into chromatin. Nucleosomes are the fundamental unit. Nucleosomes are coiled into 30 nanometer fibers. Nucleosome organization was demonstrated by the digestion of chromatin with varying concentrations of DNAs. That's a very important experiment. You should make sure you understand that well. Radial loops attached to the DNA scaffold. Chromosomes form domains in the nucleus and attach to the nuclear matrix. And multiple proteins organize the chromatin into metaphase chromosomes during cell division.